During organogenesis, literally organ formation, the three embryonic germ layers formed during gastrulation differentiate into tissues, groups of cells with similar function, organs, several tissues that perform one or more specialized functions, and organ systems, group of organs that work together to form a body function. Remarkably, organs grow to a certain size and shape, but do not grow too large or small. Clearly, the embryo cells need to properly communicate with each other so that they can proliferate, grow, undergo rearrangements, and differentiate into all tissues and organs of the embryo. Histogeny, literally tissue formation, is the differentiation of the germ layers to form specialized tissues, Fig 9.1. Adult vertebrates have four major types of tissues: 1. Epithelial tissue, epithelia; 2. Connective tissue; 3. Muscle tissue; and 4. Nervous tissue. Epithelia are composed of closely adjoined cells that act as sheets to cover surfaces or line body cavities, ducts, lymph vessels, and blood vessels. Many connective tissues act as supportive tissue that anchors organs and joins tissues. Embryonic connective tissue, called mesenchyme, derived from mesoderm, has the potential to differentiate into adult connective tissues that include cartilage, bone, adipose tissue, blood, and fibrous connective tissue. Muscle tissue is derived from mesoderm, and nervous tissue from neural ectoderm. Two or more germ layers can combine to form a single organ with an outside layer derived from one germ layer and an inside layer derived from a different germ layer. For example, the inner lining of the digestive tube is derived from endoderm and becomes surrounded by mesoderm-derived layers of smooth muscle. Also during organogenesis, some cells migrate large distances to reach their final location. For example, avian and mammalian germ cells migrate from the area pellucida or yolk sac where they originate, through the hindgut to the genital ridge, which is the primordium, rudiment, of the gonad. The vertebrate embryo acquires bilateral symmetry with three distinct axes: 1. Anterior-posterior axis or cranial-caudal axis; 2. Dorsal-ventral axis; and 3. Left-right axis. The appearance of the primitive streak during gastrulation in birds and mammals allows one to discern the cranial-caudal and left-right axes of the embryo. During organogenesis, the embryo develops a tube within a tube body plan in that it acquires a neural tube, digestive tube, respiratory tube, and heart tube with blood vessels. Developmental biologists use several vertebrates to study organogenesis, Table 1.1. Although there are differences in exactly how vertebrates develop, this chapter highlights their similarities by discussing common derivatives of the germ layers. The subsequent chapters discuss specific features of organogenesis of the frog, Chapter 10, Chicken, Chapter 11, Pig, Chapter 1-2, and Human, Chapter 13. Tables in Appendix A highlight the appearance of specific features in model organisms. Amniotes are organisms that form an amnion as one of their extraembryonic layers, and this includes reptiles, birds, and mammals. Ectoderm derivatives. Major organ systems form from ectoderm: nervous system and incumbent system. The nervous system is divided into the central nervous system and peripheral nervous system. Central nervous system, CNS. The CNS, brain and spinal cord, is derived from the neural tube that formed during neurulation, Chapter 8. At first, the neural tube is straight, but soon bends as it develops dilations and constrictions. Initially, three major brain regions are visible in the cranial neural tube: prosencephalon, forebrain; rostrally, mxencephalon, midbrain, in the middle; and rhombocephalon, hindbrain, caudally. Soon these brain regions of the neural tube develop into 5 distinct regions, with each differentiating into specific structures found in the adult, Fig 9.2. Each brain region has a corresponding cavity, the future brain ventricles. Each brain region performs specific functions. The telencephalon region of the prosencephalon is the most rostral, and will become the cerebrum, functions in association, hippocampus, functions in memory, and olfactory lobes, function in smell. The diencephalon region of the prosencephalon is very distinct early in organogenesis because it develops paired swellings, called the optic primordia of the eyes, that become the optic MP, as seen in Fig 9.2. A small area of the floor, ventral side, of the diencephalon evaginates to form a small pouch called the infundibulum that develops into the posterior pituitary gland, neurohypophysis. The anterior pituitary, adenohypophysis, develops from a nearby ectodermal invagination of the roof of the oral region called Rathke's pouch. A small area of the roof of the diencephalon evaginates to form the epiphysis, which develops into the adult pineal gland, epithalamus. The diencephalon also gives rise to the thalamus, and the hypothalamus which regulates temperature, breathing, and sleep. The mesencephalon, which has thick walls, roof, dorsal, and floor, ventral, is highly dilated and appears oval in transverse sections. It carries fiber tracts between the forebrain and hindbrain. The mesencephalon is the fourth brain region and develops into the cerebellum and pons. It functions to coordinate muscle movements. The myelencephalon is most caudal and is characterized by its thin roof and thick lateral walls and floor, Figs 9.3. 9.4. It becomes the medulla and functions as a reflex center of involuntary activities. The metencephalon and myelencephalon are difficult to distinguish from each other at early stages of development and are collectively called the rhombencephalon. Posterior to the myelencephalon is the pinal cord, which lies dorsal to the notochord. There is no distinct boundary between the myelencephalon and the spinal cord in the early embryo. The spinal cord is also characterized by a thin roof and thick ventral walls, but can be distinguished from the myelencephalon by a thin floor and paired spinal ganglia, or somates, immediately lying outside it, Figs 9.3 to 9.5. Ganglia, which are mainly derived from neural crest cells, are clusters of nerve cell bodies with a similar function. The cavity of the spinal cord is the spinal canal, Figs 9.4, 9.5. The wall of the spinal cord differentiates into three layers, Fig 9.5. The innermost layer that lines the spinal canal is the ependymal layer, ventricular zone, which appears dark since it contains many cell nuclei. The next layer is the lighter colored mantle layer, intermediate zone, that contains neuroblasts, embryonic nerve cells, and spongioblasts, supporting cells. The outermost layer is the marginal layer, marginal zone, that is light staining since it is white matter, contains many axon fibers and few nuclei. The ventral part of the mantle layer dilates to form the motor horn of the spinal cord. The dorsal half of the spinal cord is the alar plate that is demarcated from the basal plate, ventral half, by the sulcus limit. Ms, which is a slight depression that runs the length of each side of the spinal cord. The dorsal half of the spinal cord carries sensory information, and its ventral half carries motor information. Peripheral nervous system, PNs. The PNs integrates peripheral areas of the body with the CNS. The PNs is divided into cranial nerves that arise from the brain, and spinal nerves that emanate from the spinal cord. Spinal nerves are derived from the spinal cord and or neural crest cells. Spinal nerves have a dorsal root that carries sensory, afferent, information to the spinal cord, and a ventral root, motor horn, that carries motor, efferent, information from the spinal cord, Fig 9.5. The dorsal root ganglion, spinal ganglion, is a swelling of the dorsal root that contains clusters of sensory nerve cell bodies, the axons of which contribute to the spinal nerve. Paired spinal ganglia are visible in the neck and trunk of well-developed vertebrate embryos, Figs 9.5, 9.6. Several pairs of cranial nerves emerge from the ventral surface of the brain to innervate structures in the head, neck, and trunk of the embryo, Fig 9.6, Table 9.1. Frogs have 10 pairs of cranial nerves, chickens 11 pairs, and 12 pairs are in mice, pigs, and humans. The cranial nerves are designated by Roman numerals which indicate the order in which the nerves are positioned from the anterior to the posterior of the brain. Some cranial nerves are associated with special senses and are sensory, afferent, only. Others carry motor information, efferent, necessary for muscle contraction. Nerves that carry both motor and sensory fibers are called mixed nerves. The cell bodies of sensory fibers are located in cranial ganglia derived from neural crest cells. Special senses. The sensory organs, eyes, ears, nose, arise in part from paired plate-like ectodermal thickenings called placodes. The placodes invaginate to form vesicles which interact with the appropriate area of the neural tube to carry sensory information. 3 types of placolae form. The olfactory placodes are the most rostral and lie outside the prosencephalon, Fig 9.4a. They form nasal pits which line the nasal cavity and become associated with the olfactory ganglion. Each lemplacode lies outside the diencephalon and forms a lens vesicle that differentiates into the lens of the eye, Figs 9.6 to 9.8. The paired optic vesicles from the diencephalon give rise to the optic cups which develop into the retina of the eye, Fig 9.4. The auditory, otic, placodes which lie outside the myelencephalon invaginate to form the auditory vesicles, otocysts, that differentiate into the inner ear and become associated with the acoustic ganglion, Fig 9.9. The middle ear forms from an invagination of the pharyngeal wall at the level of the first pharyngeal pouch that becomes the eustachian tube. The mammalian pinna, lobe, of the external ear develops from swellings of the mandibular and hyoid pharyngeal arches. Integumentary system. Adult integument, skin, is composed of an outer epidermis, derived from ectoderm, and an inner dermis, derived from mesoderm. The integument of the embryo begins as a single-cell thick layer of skin ectode cream, Fig 9.4. The skin ectoderm differentiates into a temporary outer protective periderm, and an actively proliferating inner layer called the basal layer stratum germinativum, which later differentiates into specific cells of the epidermis. The dermis and underlying connective tissue is derived from mesoderm. 3 cell types invade the epidermis: melanoblasts, Langerhans cells, and Merkel cells. Melanoblasts are derived from the neural crest and differentiate into melanocytes which produce melanin pigment. Langerhans cells arise from bone marrow to populate the epidermis and function in the immune system. Merkel cells derived from the neural crest associate with free nerve endings in the skin and become mechanoreceptors. As the embryo develops, the epidermis may further differentiate to form scales, feathers, or hair. Glandular tissue may penetrate the epidermis. Endoderm derivatives. The embryonic gut is a simple tube of endoderm that gives rise to the pharynx and alimentary canal and associated digestive glands. In the frog, the archenteron elongates and develops into regions of the primitive gut, the anterior foregut, middle midgut, and posterior hindgut, Fig 8.21. In birds and mammals, the primitive gut lies open over the yolk sac, and forms by pinching in of body folds, Fig 8.25. The 3 major body folds include, 1. Head fold which forms the foregut that opens caudally as the cranial intestinal portal, 2. Lateral body folds which move toward each other to form the midgut, and 3. Tail fold which forms the hindgut that opens cranially as the caudal intestinal portal. At first, the digestive tube of amniotes is closed to the external environment, Fig 9.10. Invaginations of surface ectoderm meet the endodermal gut at the rostral and caudal ends of the body. Anteriorly, the foregut meets an invagination of the epidermis called the stomatocum, forming a thin ectodermal plate called the oral plate, oral membrane. The oral plate ruptures to form the margins of the mouth. The stomaterne becomes the BM-cal cavity, cavity of the mouth. At the posterior end of the digestive tube, the hindgut meets an invagination of the epidermis called the proctodeum, forming the thin cloaca membrane, anal plate, that ruptures to form the anus. The proctodeum becomes the cloaca. The primitive gut forms from endoderm, which lines 2 tubes in the embryo, the digestive tube, and the respiratory tube. The digestive tube undergoes dilations and constrictions to form the digestive system, Fig 9:10. Splanchnic mesoderm of the lateral plate mesoderm. Surrounds the endoderm of the primitive gut and gives rise to its musculature. Different portions of the digestive rube bud off into diverticula, outgrowths, forming the liver, gallbladder, pancreas, and respiratory tube. The respiratory tube bifurcates into right and left lung buds, which grow and further subdivide into the lobes of the lungs. The area of the digestive rube that is anterior to the respiratory tube evagination is the pharynx, which is derived from the foregut. Posterior to the pharynx, the digestive tube differentiates into derivatives of the foregut, esophagus, stomach, midgut, small intestine, and hindguer, large intestine, racrum. The pharynx dilates, while the esophagus remains relatively narrow. The stomach also dilates, and the intestines significantly elongate into coiled loops. In chickens and mammals, the connection of the digestive tube to the yolk sac is eventually lost. In mammals, the umbilical hernia forms a loop of the intestine within the umbilical cord and is resorbed before birth, see Fig 9.19. The pharyngeal area of the foregut expands laterally into a series of 5 paired evaginations called pharyngeal pouches, visceral or bronchial pouches, Figs 9.11-9.13. The pharyngeal pouches meet ectodermal invaginations called pharin 1,1 eel grooves, depressions, forming the closing plates, partitions. When the closing plates rupture in fishes and larval amphibians, pharyngeal clefts, slits, form which function as gill slits. In higher vertebrates, pharyngeal slits do not form, or if they do, they are not functional and are soon covered over. Derivatives of the pharyngeal pouches are shown in Table 9.2. 5 pairs of pharyngeal pouches form in vertebrates, but some become rudimentary. 4 pairs persist in birds and mammals. Between the pharyngeal pouches and grooves are the pharyngeal arches, Figs 9.6, 9.11 to 9.13. The first pharyngeal arch splits into 2 parts, the maxillary process, future upper jaw, and the mandibular process, future lower jaw. The second pharyngeal arch is called the hyoid arch. Up to 6 pharyngeal arches form, but the 5th becomes rudimentary in humans. Within each pharyngeal arch develops a skeletal element, bone or cartilage, nerve, cranial nerve, muscle, and blood vessel, Table 9.1. Mesoderm derivatives. Esoderm of vertebrate embryos differentiates into 5 specific types, Fig 9.14-1, Chonlamco LCRM, Noto. Cord, 2, Pair-chordal plate mxid cream or head MCC and CHYMC, connective tissue and musculature of face, 3, Pharyngeal mxid cream, somits, 4, Intim 1 diaceme xoderm, urogenital system, and 5, lateral plate mesoderm, circulatory system, lining of body cavities, bones of limbs, which splits into somatic mesoderm, dorsal, and splanchnic memderm. Ventral head mesenchyme is shown in figures 9.4, 9.8, 9.9, 9. 13. The somatic mesoderm and ectoderm form a double-layered membrane called the somtoplerk, which forms an extension of the body wall that gives rise to the amnion and chorion in birds and mammals. The splanchnic mesoderm and endoderm form a double-layered membrane called the splanchnoplacure that generates the gut wall, yolk sac, and allantois, embryonic bladder. The space between the somatoplure and splanchnoplure is the cochlum, Fig 9.14. Spermatogenesis. During this process, somates, blocks of mesoderm tissue, are formed from paraxial mesoderm, which lies on each lateral side of the neural tube/notochord, Figs 9.3, 9.4, 9.9, 9.13, 9.14. The number of somates varies in different species. Each somite further differentiates into 3 regions, the dermatome, myotome, and sclerotome, Figs 9.4, 9.13. The dermatome is located beneath the epidermis in the dorsolateral region of the somite. It gives rise to the dermis of the back, and becomes connective tissue of the dorsal skin. The myotome is the middle layer of the somite and differentiates into myolasts, embryonic muscle cells, that form deep muscles of the back and body wall, and muscles of the rib cage, limbs, and tongue. The sterotome is located ventromedially in the somite and differentiates into CHCM dendrocytes, cartilage-producing cells, that form cartilage of the vertebrae, back, and ribs, which is later invaded by bone. Chordiogenesis. This process refers to formation of the heart. The vertebrate heart forms from two patches of splanchnic mesoderm, Figs 8.28, 8.37. Each patch of splanchnic mesoderm hollows out to form an endothelial line tube called the endocardial tube. The two tubes fuse beneath the foregut to form a single heart tube, Fig 8.52. The embryonic heart has four segments: 1. Sinus venosus, posterior end; 2. Atrium; 3. Ventricle; and 4. Bulbus cordis, anterior end, Fig 9.15. The sinus venosus and atrium receive blood, while the ventricle and bulbus cordis are involved in pumping blood from the heart. The heart is initially straight, but eventually curls on itself and develops constrictions and dilations, that form the atrium and ventricle, Figs 9.3, 9.15. The atrium divides into a right and left chamber. The ventricle of the frog is undivided, whereas the avian and mammalian ventricle divides into a right and left side to form a four-chambered heart. The walls of the heart are composed of three layers. The inner layer is the endocardium, which is surrounded by the middle layer, the myocardium, Fig 9.9. The outer layer develops later and is called the pericardium, visceral pericardium. The heart is surrounded by a thin layer of somatic mesoderm called parietal pericardium, which forms the future pericardia sac and encloses the heart within the pericardia cavity. Blood flows through the embryonic heart as follows, Fig 9.15. Blood enters the 1. Sinus venosus, formed by the junctions of the inferior vena cava and common cardinal veins. 2. Right atrium, passes through the foramen ovale, opening, directly into the 3. Left atrium. 4. Left ventricle. Hulha cordis, bulbus arteriosus, conus arteriosus, and truncus arteriosus, ventral aorta, aortic sac, which gives off the aortic arches and external carotid artery. In chickens and mammals, a small amount of blood enters the right ventricle from the right atrium. It then flows to the pulmonary trunk, and either bypasses the lungs through the ductus arteriosus, a connection of the left pulmonary artery to the aorta, or enters the pulmonary arteries, lungs if they have developed, pulmonary veins, left atrium, left ventricle, and out the aorta. Aortic arches. 6 paired aortic arches emerge from the truncus arteriosus and run dorsally through the pharyngeal arches to the dorsal aorta, Figs 9.15, 9.16. Depending on the species, certain aortic arches, 1, 2, and 5, become transitory as development proceeds. The remaining aortic arches are transformed into adult structures, Fig 9.17. Vasculogenesis. This is the formation of blood vessels which form from lateral plate mesoderm. In the yolk sac area, or area opaca in 18-hour chick embryos, splanchnic mesoderm cells differentiate into hemangioblasts that form clusters of cells called blood islands. The outer cells of the blood islands become angioblasts, precursors of blood vessels, whereas the inner cells become hematopoietic stem cells that give rise to all types of blood cells. Once primitive blood vessels form, they become remodeled by angiogenesis to become arteries, arterioles, capillaries, venules, and veins. Arterioles is one of the major arteries that runs along the anterior/posterior axis in the embryo is the dorsal aorta, Figs 9.15, 9.18. Cranially and caudally it is a paired vessel called the dorsal aorta, but in the middle of the embryo the dorsal aorta is fused into a single vessel called the descending aorta. Three major arterial systems come from the dorsal aorta. 1. The paired internal carotid arteries are anterior extensions of the paired dorsal aortae at the level of the first somite that supply the brain and head. 2. The vitelline artery emerges ventrally at mid-body level to branch over the yolk in birds and mammals. And 3. Emerging from the caudal dorsal aorta are the allantoic arteries, which are found on either side of the allantois, future urinary bladder, and function in gaseous exchange. In mammals, the allantoic arteries become the umbilical arteries. Other branches that lead off the dorsal aorta include: 1. External carotid arteries, which supply the mandibular process of the first pharyngeal arch. 2. Intersegmental arteries, which branch dorsolaterally between somites. 3. Pulmonary arteries that supply the lungs. 4. Brachia arteries, which supply the forelimbs. Mesenteric artery, which supplies the developing gut. Renal arteries, glomi, which supply the embryonic kidneys. Iliac arteries, which branch at the level of the posterior limb buds. And 8. Caudal artery, which supplies the tail. Figs 915, 918. Veins. The embryo has two major venous systems, Figs 915, 9. 1-8. The cardinal system, which drains the head, neck, and body wall into the sinus venosus, and the telline system, which drains the yolk sac, gut, and its derivatives. In mammals, a third venous system called the umbilical system carries oxygenated blood and nutrients from the placenta into the embryo through the umbilical omphallomesenteric veins. The cardinal system is composed of cranial cardinal veins, also called precardinals, and anterior cardinal veins, future subclavian vein, that carry blood from the head to the common cardinal wimb, which empty into the sinus venosus. The caudal cardinal vesim, postcardinals, posterior cardinal veins, carry blood from the posterior body wall and kidneys. Pulmonary wind drain the lungs and bring blood to the right atrium. Vit limb, vein bring nutrients from the yolk sac to the embryonic liver in the hepatic portal veins, which become broken up by liver sinusoids, blood-filled spaces in the liver. The hepatic vein carries blood from the liver sinusoids to the inferior vena cava just before it enters the sinus venosus. Urogenital system. Intermediate mesoderm differentiates into the urogenital system, which generates the kidneys, gonads, and associated duct systems. Depending on the species, 3 pairs of embryonic kidneys develop. The pronephros, pronephric kidney, is the primary functioning embryonic kidney in fish and frogs. It is the permanent kidney in fish, but degenerates in frogs, and amniotes, birds and mammals. The mesonephros, mesonephric kidney, is the second kidney to form in frogs and amniotes, Figs 9.10, 914, 9.19. In frog, chicken, and pig embryos, the mesonephros functions in urine production, but in mice and humans it does not. It is permanent in the frog. In female amniotes, the mesonephros degenerates completely, but in males, tubules of the mesonephros are permanent and form efferent ducts of the male reproductive system. The permanent kidney of amniotes is the metanephros, metanephric kidney. The gonads develop in close association with the mesonephric kidneys. While the kidneys develop early and are visible at the embryonic stages presented in this atlas, the gonads develop later, 11 mm frog tadpole, 8-day chick, week 8 in humans. Prior to this, a rudiment of the gonads called the genital ridge, gonadal ridge, forms from the germinal epithelium next to the developing kidneys, Fig 9.19. The primordial germ cells, which migrate from the yolk sac splanchnic mesoderm or area pellucida, will later populate the genital ridge, which thickens and elongates into medullary, internal, sex cords that form seminiferous tubules in males. In females, the germ cells migrate to cortical sex cords that eventually form follicles. Body cavities. The embryonic chylorn becomes subdivided to form the adult body cavities. In fishes, amphibians, and most reptiles, the chylome is divided into the pericardia, cavity that houses the heart, and the pleuroperitoneal cavity that contains most of the other viscera. In mammals, the lateral plate mesoderm lines three major body cavities: 1. Paired pleural cavities that house the lung buds, future lungs; 2. A single pericardia, cavity that contains the heart; and 3. The peritoneal cavity that houses the abdominal organs. Flexion and torsion. During organogenesis, the embryo may undergo flexion, bending along the anterior-posterior axis, and/or torsion, twisting in the left-right axis. Chick embryos undergo flexion and torsion, giving them a hook or backward question mark shape, Figs 9.3, 9.12. Mammalian embryos undergo flex-ion, forming a C shape. As an embryo elongates, it may curl upon itself. This is readily seen in later stage fish embryos. Before hatching, the embryo is coiled on itself with the tail extending well past the head, Fig 9.20. By comparison, frog embryos are only slightly curved prior to hatching, Fig 9.7. The curled nature of vertebrate embryos makes interpreting sections on slides challenging. For example, the first transverse sections of a serial section series of a C-shaped 10 mm pig embryo contain a caudal brain region, metencephalon and/or myelencephalon, rather than the rostral-most brain region, telencephalon, which is located at the tip of the letter C, Figs 2.14, 9.6. Moreover, the very first transverse section of a 48-hour chick embryo serial section series usually contains the mesencephalon or metencephalon rather than the telencephalon, which at this stage lies close to the heart, Fig 9.3. Limb development. Limbs are outgrowths of the body wall of the embryo and are formed from a mesenchyme core that is covered with ectoderm. Two pairs of limb buds form in most vertebrates, and the two limbs of each pair are mirror images of each other, Figs 9.12, 9.21. The anterior pair is called forelimb buds, wing buds, and the posterior pair is rhe hind limb buds, leg buds. When comparing species, limb buds form at different somite levels. However, the first pair of limb buds typically form at the level of the first somite in amphibians, birds, and mammals. A limb field is an area of the embryo that is capable of forming a limb. Lateral plate mesoderm in the limb field induces myoblasts, embryonic muscle cells, from the somates to enter the limb bud and become muscles of the limb, whereas the somatic mesoderm enters the limb bud to form skeletal precursors. The distal tip of the limb bud acquires a thickened raised edge of ectoderm called the apical ectodermal ridge, AIR, that runs along the anteroposterior margin of the limb bud, Fig 9.21. The AIR is an important signaling center that plays a role in reciprocal inductions of the limb bud. The AIR is absent from urodels, but is present in limb buds of amniotes. In limbless lizards and snakes, limb buds may initiate but fail to maintain the AIR, or it doesn't form, and limbs fail to develop. In many species, specific cells at the distal tip, farthest end, of the limb bud die by apoptosis, programmed cell death, allowing digits to form. Metencephalon rather than the telencephalon, which at this stage lies close to the heart, Fig 9.3. Limb development. T. Limbs are outgrowths of the body wall of the embryo and are formed from a mesenchyme core that is covered with ectoderm. Two pairs of limb buds form in most vertebrates, and the two limbs of each pair are mirror images of each other, Figs 9.12, 9.21. The anterior pair is called forelimb buds, wing buds, and the posterior pair is the hind limbite buds, leg buds. When comparing species, limb buds form at different somite levels. However, the first pair of limb buds typically form at the level of the first somite in amphibians, birds, and mammals. A limb field is an area of the embryo that is capable of forming a limb. Lateral plate mesoderm in the limb field induces myoblasts, embryonic muscle cells, from the omites to enter the limb bud and become muscles of the limb, whereas the somatic mesoderm enters the limb bud to form skeletal precursors. The distal tip of the limb bud acquires a thickened raised edge of ectoderm called the apical ectodermal ridge, AIR, that runs along the anteroposterior margin of the limb bud, Fig 9.21. The AIR is an important signaling center that plays a role in reciprocal inductions of the limb bud. The AIR is absent from urodels, but is present in limb buds of amniotes. In limbless lizards and snakes, limb buds may initiate but fail to maintain the AIR, or it doesn't form, and limbs fail to develop. In many species, specific cells at the distal tip, farthest end, of the limb bud die by apoptosis, programmed cell death, allowing digits to form. 1. Jo. The frog is an excellent vertebrate model to study organogenesis because of its relatively simple embryology, and small size. It shares many features with the lancelet, branchiostoma, but is more advanced. Analysis of amphibian organogenesis will aid in understanding the more complex development of birds and mammals. The previous chapters have followed the development of the fertilized frog oocyte to the Neurula stage. In this chapter, specific stages of frog, Ranapapiens, development were chosen so that features of the organ systems can be identified. Specific post-Neurula stages are identified according to the embryo's length in millimeters in addition to the Shumway staging series, Table A8. External morphology. After neurulation, the frog embryo elongates and forms head, neck, trunk, and tail regions. These regions are first visible at stages 17, tail bud, and 18, 4 millimeters, Figs 10.1. 10.4. The tail bud significantly elongates to form a tail with dorsal and ventral fins. The shape of the embryo changes from spherical to elongated. The embryo's anterior enlarges, and its posterior narrows laterally so that the tail is very thin as compared to the rest of the body, Figs 10.10, 10.23, 10.39, 10.53. The back changes from a concave curve to a straight line. The embryo develops prominent bulges on its external surface. These are first seen in the neurula, stages 14 to 16. The sense plate forms the first pharyngeal arches, Figs 8.13, 8.20, 10.22c. The gill plates, which are swellings immediately posterior to the sense plate, develop plume-like external gills visible at stage 1 and 9/20, 5 to 6 millimeters, Figs 10.1, 10.10 to 10.12, 10.24, 10.25. The gills function in respiration and are highly vascularized. The external gills become covered by the operculum, which is a growth of skin from the second pharyngeal arches, that internalizes the gills within the opercular chamber, gill chamber or parabranchial chamber, Figs 10.39, 10.52. While the right gills are completely covered by the operculum at stage 25, 1 mm, the spiroch, small opening on the left side, remains open until metamorphosis, Figs 10.53, 10.54. During respiration, water taken in through the mouth passes over the gills in the opercular chamber and is expelled through the spiracle. Other external swellings of the embryo are the eyes, Figs 9.8, 10.1, 10.3, 10.4, 10.12, 10.25, 10.39, 10.54. The pronephric region is a bulge located on each side behind the neck, and contains the pronephros, which is the first embryonic kidney, Figs 10.1, 10.4, 10.11, 10.24. Also on each side of the trunk are paired chevron-shaped myotomes that form muscles derived from somates, Figs 10.23, 10.24. Contractions of the myotomes aid during hatching and enable the young tadpole to swim using undulating tail movements. On the ventral surface of the head, a prominent, horseshoe-shaped adhesive gland with oral suckers develops, allowing the tadpole to adhere to surfaces after hatching. Figs 10.1 to 10.4, 10.10 to 10.12, 10.23 to 10.25, 10.39, 10.55. On the anterior side of the head, the paired olfactory placodes invaginate forming nasal pits, Figs 10.3, 10.4, 10.10 to 10.12, 10.23, 10.39, 10.54. The mouth opens at stage 21, 7 mm, however, a plug of endoderm located in the esophageal region prevents the embryo from digesting food until stage 23, 9 mm, Figs 10.25, 10.39. Until then, the yolk in the intestines is the sole food source. The mouth has lips that surround 3 to 4 rows of teeth used in scraping vegetation for food, Fig 10.55. Integument. Internal morphology. During stage 15, neural groove, the epidermis, integument, develops cilia that form prominent tufts. The ciliary tufts allow the embryo to rotate within the jelly capsule prior to hatching, Figs 10.3, 10.9. Eventually the cilia disappear, but persist on the tail until stage 25, 11 mm. The adhesive gland arises from the epidermis in the head at stage 14, neural fold, and its oral suckers begin to degenerate after stage 20, 6 mm, Figs 10.5 to 10.9, 10.14, 10.16, 10.19. 10.22 BE, 10.27 to 10.29, 10.31 G, 10.38 DE, 10.44, 10.52 E. Skin pigment is derived from melanin, black pigment, of the oocyte, and melanophores, pigment cells of the skin which contain organelles called melanosomes that house melanin granules. Melanophores are readily seen in the anterior of the stage 24, 10 mm, tadpole, Fig 1052 AD. Central and peripheral nervous systems. The central nervous system is visible as a hollow elongated neural tube running most of the length of the neurula. The dilations of the brain that form the prosencephalon, mesencephalon, and rhombencephalon are first visible in the neurula, stage 16. By the tail bud stage, stage 1-7, the anterior of the neural tube is curved ventrally in a cranial flexure, Figs 10.7b, 10.29. At stage 21, 7 mm, the walls of the central nervous system have differentiated into the thin inner ependymal layer, broad middle mantle layer, gray matter, and fibrous outer marginal layer, white matter, which are more easily identified at stage 24, 10 mm, Figs 10.48, 10.52. Also at this stage, the meninges, covering of the central nervous system, are first visible, Fig 10.52e. Just before hatching stage 20, 6 mm, the telencephalon and diencephalon can be discerned, Fig 10.16. By stage 24, 10 mm, the telencephalon bulges into envolateral halves, the future cerebral hemispheres that are separated by the lamina terminalis, a central, unexpanded region. Of the telencephalon, Fig 10.52 AB. Each telencephalic vesicle contains a lateral ventricle, tclococal, connected to the diacocal, cavity of the diencephalon that forms the adult third brain ventricle, by the foramen of Monro. The olfactory nerves form on the ventral side of the telencephalon. The roof of the diencephalon has a profuse capillary blood supply called the anterior choroid plexus, which is located anterior to the epiphysis, future pineal gland. Figs 10.38a, 10.43, 10.48, 10.52 CD. The infundibulum which forms the neurohypophysis, posterior pituitary, evaginates from the floor of the diencephalon toward the adenohypophysis, anterior pituitary, from the pharynx. Figs 10.9d, 10.29, 10.34, 10.43. The diencephalon also gives rise to the optic vesicles, each with their own cavity, the optic oval, Fig 10.9b. The second cranial nerves originate from the retina derived from the optic vesicle. The mesencephalon has two grooves on its dorsal surface, which separate it anteriorly from the diencephalon and posteriorly from the rhombencephalon, Figs 10.16, 10.29. Ventrally, the posterior end of the mesencephalon is marked by the posterior tubercle. The mesococcal cavity of the mesencephalon narrows to form the cerebral aqueduct, aqueduct of Sylvius, connecting the diaceal with the rhombococcal cavity of the rhombencephalon. Two cranial nerves are associated with the mesencephalon, oculomotor and trochlear. The metencephalon and myelencephalon are not distinguishable from each other at the stages shown, and are collectively called the rhombencephalon, Fig 10.16. The rhombocoel forms the adult fourth brain ventricle. The thin roof of the myelencephalon has a rich capillary blood supply that forms the posterior choroid plexus, Fig 10.52 Fg. All the cranial nerves except the first four are associated with the myelencephalon. The spinal cord is continuous with the rhombencephalon and terminates prior to the tip of the tail. It has thick lateral walls and a thin roof and floor that appear oval in anterior transverse sections, and small and round posteriorly. This is especially evident in the 10 mm tadpole, stage 24, Figs 1052 Ks. Paired spinal ganglia are visible lateral to the spinal cord at this stage, Fig 10.52 K. While 40 pairs of spinal nerves form in the tadpole, only 10 persist in the adult. Special sense organs. Development of the sensory organs in the 7 to 10 mm larvae, stages 21-24, has advanced significantly from the placode stages. The olfactory placodes appear at stage 16, Neurula, have invaginated to form deep olfactory pits, the walls of which have thickened to form the nasal organ, Figs 10.9a, 10.35, 10.38a. At stage 24, 10 mm, a dorsolateral evagination of the olfactory pits forms the vomeronasal organ, Jacobson's organ, lateral appendix, that functions to sense food in the oral cavity, Fig 10.52 BD. The eyes develop from optic vesicles, visible at stage 16, Neurula, from the diencephalon as described in Chapter 9, Figs 10.7a, 10.9 BD, 10.20b, 10.22b, 10.33, 10.34. 10.38 BD, 10.44, 10.48, 10.52 pound. The lens develops from the lens placode visible at stage 18, 4 mm, Fig 10.7a. At stage 24, 10 mm, the sensory layer of the optic cup has differentiated into distinct layers, Figs 10.44, 10.48, 10 pounds and 52 pence. At this stage, the lens contains transparent 1 cm fibers surrounded by a 1-cell thick layer called the LCN, epithelium. The cornea, outermost layer covering the eye, is composed of outer ectoderm and a thin mesodermal layer visible between the ectoderm and the lens, Figs 10.48, 10 pounds and 52 pence. Each otic vesicle, derived from the otic placode seen at stage 15, neural groove, elongates before it develops into the adult inner ear. At stage 24, 10 mm, an oblique PA partition divides the otic vesicle into a dorsal inner chamber, which becomes the utriculus and a ventral sanulus, Fig. 10.42, 10.44, 10.48, 10.52 fH. The utriculus will give rise to the semicircular canals that function in equilibrium. The SACCULUS generates the lagina, cochlea, which functions in hearing. In addition to the nose, eyes, and ears, the tadpole develops the lateral line organs, one on each side, from cells that contribute to the 10th cranial nerve. The lateral line organs are aquatic larval sense organs that respond to changes in water currents. They disappear at metamorphosis. Digestive and respiratory systems. The relationships of the structures of the digestive and respiratory systems are shown in Figure 10.41. The mouth forms from the stomodium, which is closed by an oral plate until stage 21, 7 mm, when the oral plate ruptures, Figs 10.7b, 10.16, 10.29, 10.43. At the neurula stage, the digestive system consists of foregut, midgut, and hindgut. The foregut differentiates into the oral cavity, pharynx, esophagus, stomach, and duodenum, Figs 10.42, 10.43, 10.52. The pharynx becomes conical in shape, Figs 10.7, 10.9d. Pharyngeal arches, pouches, and clefts, slits, characterize the pharynx, Figs 10.20b, 10.22c, 10.35, 10.38. The third-fifth pharyngeal arches form external gills, Fig 10.35. Aortic arches are visible within the pharyngeal arches and provide blood vessels to the gills, Fig 10.34, 10.44. In older tadpoles, stage 24, 10 mm, velar plates grow from the dorsolateral and ventral sides of the pharynx and function to prevent food from clogging the gills, Fig 10.43. A single evagination in the floor of the pharynx, just anterior to the heart, develops into a darkly staining thyroid gland, Figs 10.43, 10 pounds and 52 pence. In the posterior of the pharynx, the laryngotracheal groove gives rise to the trachea and two thick-walled, oval lung buds visible at stage 24, 10 mm, Fig 10.52 Hj. The pharynx is discerned from the esophagus by the presence of the glottis, which is the opening from the pharynx into the trachea, Figs 10.43, 10.52 g. The trachea, lung buds, and digestive organs are housed in the pleurocrotonchal cavity, which is a large chylome, Fig 10.43, 10.521 j. The foregut also generates the liver, which first appears as a diverticulum in the neural fold stage, stage 15. The liver has loose cords which are surrounded by sinusoids, spaces filled with blood, Figs 10.43, 10.521 k. The gallbladder forms from the posterior region of the liver diverticulum. The bile duct drains the contents of the gallbladder into the duodenum, Fig 10.52 k, L. The pancreas is derived from three diverticula that form near the liver rudiment, Figs 10.43, 10.52 L. The liver, gallbladder, and pancreas are visible at stage 24, 10 mm. The stomach is distinguishable from the esophagus and duodenum not only by its dilated appearance, but also by its thick muscular and glandular wall, Figs 10.43, 10.52 k. The midgut gives rise to the majority of the intestines, which become coiled spirally in the tadpole, Fig 10.52. The intestines are full of yolk platelets from the oocyte. The hindgut becomes the rectum and its posterior region becomes the cloaca, Figs 10.38 n, 10.43, 10.52 q. The proctodeum forms the anal region, Figs 10.38 p, 10.52 r. Urogenital system. The paired pronephros are each composed of three coiled pronephric tubules associated with the posterior cardinal veins, Figs 10.9 g, 10.22 f, 10.38, 10.44. They are found on the dorsolateral side at the level of the second through fourth somates. The pronephros functions at hatching, stage 20, 6 mm. By stage 24, 10 mm, one large glomus forms, which is a highly vascularized structure, similar to a primitive glomerulus, lateral to the dorsal aorta, Fig 10.52. The pronephric tubules open into the pronephric duct, Wolffian duct, which runs from the end of the pronephros to the hindgut where it empties into the cloaca, adult urinary bladder. The pronephric duct, nephric duct, becomes the mesonephric duct in later stages, and the ureter of the adult, Fig 10.52 kp. The pronephros degenerates and is replaced by the mesonephros, adult kidney, Fig 10.520. The rudiments of the mesonephros first appear at stage 24, 10 mm. The rudiments of the gonads are not visible until stage 25, 11 mm. They form near the mesonephros from the genital ridge, and develop during metamorphosis. Skeletal and muscular systems. The first visible component of the embryonic skeleton is the notochord, which forms during gastrulation. The notochord never extends anteriorly past the adenohypophysis, and posteriorly it extends almost to the tip of the tail, Figs. 10.7, 10.43. Small blocks of cartilage are seen below the telencephalon and diencephalon, along with parts of the chondrocranium, cartilaginous skeleton of head, and a long bar of cartilage ventral to the pharynx floor, Fig. 10.52 de. The first visible muscles are the myotomes derived from the somates, Fig. 10.7. The myotomes first contract the body at stage 18, 4 mm, prior to hatching. At stage 24, 10 mm, several pharyngeal muscles have formed lateral and ventral to the pharynx, Fig. 10 pounds and 52 pence. Circulatory system. Chapter 9 discussed the frog heart and aortic arches, Figs 9.15, 9.17, which will be briefly mentioned here. Recall that the heart formed from two separate tubes that fused along their length to form a single tubular heart enclosed by the pericardia. Cavity. At stage 24, 10 mm, the heart is a slightly coiled tube that is bent to the right, Figs 10.43, 10.50, 10.52. The most anterior chamber is the bulbus cordis, which is connected to the ventral aorta, Fig 10.52f. The next chamber moving posteriorly is the thick-walled ventricle, Fig 10.52g. Next the thinner-walled atrium is positioned dorsally and will soon divide into two atria, Fig 10.52gh. The ventricle remains as a single chamber. Posterior to the atrium is the sinus venosus, which lies to the right at the anterior end of the liver, Fig 10.52h. The pericardium surrounds the pericardia. Cavity and heart, Fig 10.52g. In the early embryo, the right and left vitelline veins form around the yolk and enter the sinus venosus. As the liver grows, the left vitelline vein enlarges and becomes the hepatic vein anteriorly, between the sinus venosus and the liver, and hepatic portal vein posteriorly, between the gut and the liver, Fig 10.521. The right vitelline vein degenerates. The hepatic vein, inferior vena cava, from the liver, and the common cardinal veins from the lateral body walls drain into the sinus venosus. The posterior cardinal veins extend into the tail as the caudal vein and empty into the common cardinal veins, Fig 10.52. The anterior cardinal veins, which also drain into the common cardinal veins, become the adult jugular veins, Fig 10.52 ef. Anteriorly the dorsal aortae are paired and extend into the head as the internal carotid arteries, Figs 9.15, 10.52 f. Posterior to the aortic arches, the dorsal aortae fuse to form a single dorsal aorta which extends into the tail as the caudal artery. Metamorphosis. Dana papiens tadpoles metamorphose about 3 months. J. Rom the time the oocyte is fertilized. For some species of frog, example Ranocatesbiana, the larva stays as a tadpole through 2 summers, until it rapidly undergoes metamorphosis at the end of the 2nd summer. Several changes occur during metamorphosis. Details are beyond the scope of this atlas, however, some general changes will be mentioned here briefly. The gill slits disappear, and the lungs expand and function in respiration instead of the gills. The mesonephros becomes the functioning kidney of the adult, while the pronephros degenerates. The urinary bladder forms. Limbs develop and the tail regresses. The mouth widens and the intestines shorten. The oral suckers of the adhesive gland regress. The aortic arches are modified to the adult pattern, Fig 9.17. The diet changes from that of a herbivore to a carnivore. Rana papiens are sexually mature the 2nd spring after metamorphosis.